Welcome, curious minds, to the deep dive. Today, we're diving into a really big question. How do we actually feed everyone sustainably, with a changing climate and fewer resources?
Well, it's huge.
It really is. And the fascinating part is it's not just about more farming. It's fundamentally about farming smarter. It's about science, about technology, really revolutionizing agriculture.
Exactly. And to give you a shortcut kind of to understanding this whole complex field, we're basing this deep dive on insights from a fantastic class by Professor Guilherme da Silva Pereira at UFE.
Yeah, specifically the FIT 679 course, Biotechnology Applied to Plant Breeding.
Right. So our mission here is to pull out the key ideas from that material, share some maybe surprising facts, and give you the context you need to understand agriculture's path to, hopefully, a sustainable future.
Mm-hmm.
So let's get into it. Plant breeding. The definition often used is the art and science of improving the heredity of plants for the benefit of humankind. Sounds grand. It is pretty grand when you think about it. It's essentially about changing plants genetically, strategically, to get traits we want. But, to do that, you need to grasp two basic ideas first.
Okay, what are they?
Heredity and variation. So heredity is just how the genetic stuff, the DNA, gets passed down generation to generation.
Okay.
And that DNA holds the instructions for the plant's phenotype, basically everything you see and measure about it. Its height, yield, color, everything. It's the blueprint.
Not a blueprint passed down. And variation. That's just the differences we see.
Exactly. Differences between plants. But, and this is key, is that difference because one plant got more sun? That's environmental. Or is it actually in its genes?
Ah, right.
Mm-hmm.
And breeders only care about the genetic differences.
Well, they care intensely about genetic differences.
Yeah.
Because only genetic variation is heritable. It's the only kind you can actually select for and pass on reliably. Environmental differences, they don't stick genetically.
Okay, so you need that heritable variation. How do breeders actually, use it? You mentioned a push and pull.
Yeah. We talk about antagonistic but complementary forces. Selection needs variation. You can't select it. Everything's identical, right?
Right.
But selection reduces variation by picking the best. Heredity then makes sure those selected traits are passed on, maintaining the improvements. So you need both. Selection acts on variation. Heredity preserves the gains.
Okay, that makes sense. So if those are the building blocks, what's the actual, the cycle, the innovation cycle of breeding? What's the core idea?
The core idea is actually pretty simple. You figure out what traits you want, then you try to combine them into what we call an ideotype.
An ideotype.
Yeah, think of it as the breeder's perfect plant, the ideal blueprint they're shooting for, with all the desired characteristics combined.
So they have this vision. Then what? How do they build it?
Usually you cross the biggest plants you have, or maybe plants that have different complementary traits you want to bring together.
Okay, like mixing paints.
Sort of. And their offspring, the progenies, show new combinations. Then you have to identify the most promising ones among those.
And those promising ones, they become new varieties. Or parents for the next round.
Could be either. If they're really good, maybe a direct product. Or, more often, they become parents for the next cycle of crossing and selection.
And it all starts with the germplasm input, right? The raw material, old varieties, wild relatives.
Exactly. From gene banks, old land races, wherever you can find useful genes. Now, picking the best progenies, that's changed a lot.
How so? It used to be just looking at them in the field.
Yeah, traditional phenotypic selection. But now molecular markers are a huge deal. It's like a shortcut. Instead of waiting for a plant to grow up to see if it resists a disease, say.
You can tell earlier.
Much earlier. You look directly at its DNA for specific markers, little flags that tell you if the gene for resistance is likely there, even in a tiny seedling.
So it's like predicting the future, almost, based on the DNA.
Kind of like that, yeah. Like predicting disease risk in humans from DNA markers. For breeders, it means screening millions of plants super fast, very early. It saves enormous amounts of time and resources.
Sounds way more efficient. But the course material, it stresses that even with markers, you still need field trials before releasing a variety. Why bother if the markers look good?
Because the lab isn't the real world. DNA tells you potential, but you have to see how the plant actually performs out there. Different soils, weather patterns, pests, year after year, location after location.
Okay, the ultimate reality check.
Exactly. Field trials are non-negotiable.
And this whole cycle, you said it's never ending. Why never ending?
Because the goalposts keep moving. We always push for more yield, better quality, but then new pests show up, diseases evolve, the climate throws curveballs.
So you're always chasing new problems or higher targets.
Constantly adapting. It's a race.
Which brings up that interesting point, that paradox.
Hmm.
Selection actually decreases variation within the cycle, right? Because you're narrowing things down.
Right, you're focusing on the traits you want.
But you need more variation coming in at the start, the germplasm stage.
Precisely. And this is where biotechnology really shines. What if the variation you need just isn't there in the natural gene pool?
What do you do then?
That's when biotech can induce completely new variation. Things like mutagenesis, using radiation or chemicals, or transgenesis, putting in a gene from another species, or even precise gene editing. These tools expand the breeder's palette dramatically.
Can you give us some visual examples of this variation we're talking
Oh,
about?
sure. Think about corn. All the different kernel colors, yellow, white, blue, red, different sizes, shapes, that's variation.
Go.
Or potatoes, russets, reds, yellows, fingerlings, different skin colors, shapes, sizes, textures, all driven by genetic variation.
But the course made a great point. Just having diversity isn't the whole story. Selection is critical. The coffee example was fascinating.
Ah, yes. Coffee originates in Africa. Huge genetic diversity there.
Right. But Brazil became the world's biggest producer.
Why? Because Brazilian breeders strategically selected and bred specific types, specific idiotypes, over many years that were super productive in their environment and met market demands. Having the raw ingredients is one thing. Knowing how to combine and refine them through selection is another.
And this idea of selection, it goes way back, doesn't it?
Yeah.
To early farming, domestication.
Yeah, absolutely. Early humans saving seeds from the plants they liked best, maybe the biggest fruits, the easiest to harvest, that was selection.
Mm.
Unconscious maybe, but selection nonetheless.
And that brassica oleration example is just mind-blowing. One wild plant.
Yeah, wild mustard or something similar. And just by selecting for different parts, leaves, stems, flowers, buds, generation after generation.
I got kale, cabbage, broccoli, cauliflower, kohlrabi, Brussels sprouts.
All from that one ancestor. It shows the incredible power of sustained selection acting on variation.
It really does. So, okay, well, why does all this matter so much for us, for you listening, for the planet?
Well, because plant breeders are right on the front lines of tackling massive global challenges. Yield is always important, sure, but often the focus shifts. Like, if yield gains slow down, maybe resistance becomes the top priority.
And resistance is huge, right? Especially for cutting down on chemical use.
Definitely. Developing resistant plants is key, and climate change makes it trickier.
How so?
Warmer temperatures mean insects and diseases pop up in new places where they weren't a problem before, so breeders constantly face new threats.
On top of that, we need plants that can handle weird weather droughts, floods, climate resilience.
Exactly. And plants that use resources like water and nutrients more efficiently, getting more crop per drop, essentially.
The projections for 2050 in the source material were, well, they were pretty stark. Higher average temperatures globally.
Potential increase in crop losses to pests, maybe up to 25% globally.
Rising water demand.
A global population hitting maybe 8 billion people.
And less arable land per person to grow food on, dropping significantly.
Yeah. So the big critical question is, how do we feed all these people with less land, less water, and more environmental stress? It's a monumental challenge.
And it's not just about the total amount of food, is it? The course highlighted the inequality.
Right.
You might grow enough calories globally,
But distribution is a huge problem. Access is unequal. So you still have rising numbers of undernourished people. Recent events, pandemics, wars, they've only made that worse.
right. And it's often about access to nutritious food, not just calories. The source mentioned areas like Southeast Asia, Sub-Saharan Africa, facing big challenges with underweight children.
Absolutely. There's this curve they showed, dietary energy supply versus population increase. The gap is closing. It could cross potentially around 2040, meaning demand outstrips supply in a serious way.
Wow.
So more resilient plants, plants that need fewer inputs, resist pests better, they are absolutely crucial to preventing that crisis.
And there are other pressures too, right? Like diets changing.
Urbanization often means more demand for meat and dairy, which puts indirect pressure on feed crops.
Using crops for biofuels.
Land degradation, losing fertile soil, and just the challenge of the yield gap, the difference between what's possible to grow per acre and what farmers actually achieve. Lots of pressures.
It's clear breeders have their work cut out for them. So this brings us squarely to biotechnology. How does it help tackle these massive issues? Let's start with the definition. What is biotech?
Okay, broadly, the course defines it as a set of methods that use living beings or their parts in the development of processes and products that have an economic or social function.
So it's pretty broad. And you mentioned it's not actually new.
Not at all. Fermentation for bread, cheese, wine, beer. That's ancient biotechnology. Humans using microbes for thousands of years.
Right.
But modern biotech, that's where we see tools like molecular markers, genetic engineering, recombinant DNA, tissue culture, things that give us much more precision and power.
Like the insulin example, used to come from pigs.
Exactly. Now it's largely made using genetically engineered E. coli bacteria. Huge impact. But yeah, let's bring it back to plants.
Okay, so what were the big steps, the milestones, where biotech really started changing plant breeding?
Well, the foundations were laid by Mendel's laws back in 1866, understanding basic genetics. A huge leap was hybrid breeding, especially in maize, creating super vigorous, high-yielding plants by crossing specific parent lines.
That boosted yields massively, right?
Phenomenally. Then came mutation breeding, maybe in the 1930s onwards, using radiation or chemicals to deliberately cause random mutations, creating new variation to select from. Still used a lot, actually, especially in flowers and fruits.
And then the pace picked up even more, the genetic revolution.
Yeah, that was a real shift. With genetic engineering, gene transfer, suddenly breeders weren't just working with the variation within a species. They could bring in totally new traits.
Like taking a gene from a bacterium and putting it into a plant?
Precisely. Like the bite gene for insect resistance, that was revolutionary. You're adding functions that just didn't exist in that plant before.
Okay. And then came the tools for better selection, marker-assisted selection, MAS.
Right. MAS really took hold around the year 2000. It let breeders use those DNA markers we talked about to select plants much earlier, much more efficiently. Massive speed increase.
And genomic selection was the next step.
Yeah, maybe around 2010 onwards. Instead of looking at just a few markers linked to a trait, genomic selection looks at markers across the entire genome. It allows for much better prediction and selection, especially for complex traits controlled by many genes, like yield.
So the course highlights molecular markers and transgenics as maybe the most influential biotech milestones. Why those two specifically?
Well, because they change the game in fundamental ways. Markers revolutionize the speed and precision of selection. Transgenics revolutionize the source of variation, allowing us to introduce novel traits. And, these things often work together. You might need tissue culture, another biotech tool, to actually get the transgenic process to work efficiently.
So biotech really does two main things for breeders.
Hmm.
It helps create new variation when needed through things like mutation, transgenesis, gene editing, and it helps select the best variations much more effectively.
Exactly. Expanding the toolkit and sharpening the selection process. That's the dual role.
Okay, let's talk optimization. There's this thing called the breeder's equation. Sounds a bit intimidating.
Yeah. RHEIAH2ASPLO. Looks complex, but it basically breaks down the factors affecting how much genetic improvement you get per cycle.
So what are the parts? I is intensity.
Intensity of selection, how picky you are. H2 is heritability, how much of the variation is actually genetic. Sigma squared P is the total amount of variation you have to work with. And LL is the length of the breeding cycle, the generation interval.
Okay, so where does biotech make the biggest difference in that equation?
The biggest impact, arguably, is on L, reducing that generation interval. Molecular markers let you select super early so you can turn over generations much faster.
Shorter cycles mean faster progress.
Dramatically faster. That's a huge lever biotech pulls.
Got it. And this all fits into the modern plant breeding pipeline. What are the main stages there?
Typically 3 main phases. First is the design phase. This is where you define the target. What does the farmer need? What does the consumer want? What problem are we solving? What's the product profile?
That's setting the goals.
Exactly. Then comes the engineering phase. That's where the breeding strategy happens, choosing parents, making crosses, developing populations, selecting the best lines, putting the plan into action.
And the last phase.
The operations phase. Taking those elite lines, scaling them up, testing them rigorously for release, getting them commercialized, and then, crucially, monitoring how they do in the real world. Is the variety being adopted? Is it performing well?
And there's a feedback loop, right?
Absolutely critical. If a new variety isn't adopted, maybe the initial product profile in the design phase wasn't quite right. That feedback informs the next cycle. It's continuous improvement.
Okay. Now, in terms of bringing in new genetic variation in that engineering phase, we talked about transgenesis and editing. What else?
Well, besides those induced modifications, you still use traditional outcrossing, obviously. And sometimes variation just pops up during tissue culture. It's called somoclonal variation, often stress-induced.
Interesting. And for selection, you mentioned forward and reverse genetics.
Yeah, forward genetics is the classic approach. See a cool trait, phenotype, then hunt for the gene responsible. Reverse genetics is the opposite. Start with a gene, maybe knock it out or modify it, and then see what effect it has on the phenotype.
It's amazing comparing these modern methods. You have crossbreeding, just mixing existing traits, mutation breeding, making random changes,
Yep, like a shotgun approach,
transgenics bringing in foreign DNA,
adding completely new functions.
and genome editing making precise, targeted tweaks to existing genes.
Right, much more like using a scalpel. Different tools for different jobs, but all aiming for improvement.
Now here's a really surprising bit from the course material. Despite how different these methods seem, developing a new variety takes roughly the same amount of time.
Yeah, it's counterintuitive, isn't it? People think GMOs or gene editing must be way faster, and the initial genetic change might be. But the whole process, including all the testing, field trials, and especially the regulatory hurdles for transgenic and some edited crops, means the total time from lab bench to farmer's field often ends up being pretty similar, maybe 10, 15 years, across these different methods.
That regulatory aspect is huge.
Mm-hmm.
And the course pointed out some real contradictions globally.
Oh, definitely. It's fascinating and a bit frustrating. You have mutagenesis causing random, unpredictable mutations with radiation or chemicals, which is often completely deregulated in many places. It's been used for decades.
Okay.
But then you have highly precise genome editing, making a tiny specific change to an existing gene, and in some places, like the EU currently, it can face the same super strict regulations as introducing a foreign gene via transgenesis.
That seems inconsistent.
It is complex. Other major regions, the US, Brazil, China, Japan, Australia, others tend to take more of a case-by-case or product-based approach, looking at the final trait rather than just the method used. But Europe's stance definitely has a big global impact, even affecting, say, African countries hesitant to adopt GMOs for fear of losing export markets to the EU.
Wow. Okay, so looking forward, what's the absolute cutting edge? The course talks about the omics revolution and genomics-assisted breeding 2.0.
Right. Omics refers to all these technologies generating massive data sets. Genomics, whole genomes, transcriptomics, which genes are active, proteomics, proteins, metabolomics, metabolites.
So studying the whole system.
Exactly. Plus, we have amazing new ways to measure the phenotype precisely. Drones, sensors, cameras capturing plant growth and health in incredible detail. All this data lets us be much more targeted, much more precise in breeding.
And that leads to genomics-assisted breeding 2.0. What's new about 2.0?
It's about integrating all this data for even smarter, faster breeding. Think de novo domestication, taking wild plants and rapidly domesticating them using genomic knowledge, or speed breeding using controlled environments to slash generation times even further, or optimal contribution selection using genomics to figure out the absolute best parent combinations to maximize long-term genetic gain.
So it's faster, more precise, more informed.
That's the idea. Higher speed, greater precision.
And a really important point made was that this isn't just for the big cash crops, corn, soy, wheat, cotton.
No, thankfully. These powerful tools are increasingly being applied to what we call orphan crops.
Orphan crops.
Yeah, crops that are really important locally, maybe staples in certain regions, vital for nutrition and food security, but often ignored by the big commercial breeding programs. Think cassava, yams, sorghum, millets. Public institutions, like UFE, where this course comes from, play a crucial role in ensuring these crops also benefit from modern breeding.
That's fantastic to hear. So we've really covered a lot today, from the basics, heredity, variation, selection, all the way to gene editing and genomics.
Yeah, it's been a real deep dive. The core goals remain the same, improving quantity, quality, and sustainability of our food supply. But the tools we have now are incredibly powerful, and the challenges, as we've seen, are immense.
Absolutely, which leaves us with a final thought, something for you to chew on. If we connect all this, the huge pressure on our food systems, these incredibly powerful breeding technologies, what responsibility do we have as a global community? How do we make sure that confusing or conflicting regulations don't stop these tools from being used effectively and ensure their benefits can actually reach everyone, everywhere?